When we had lots of rain on the 19th and 20th, I was sitting drinking coffee in my three-season room thinking about going into work when my husband said, "You're not going into work today. The back alley is a stream. And by the way, they're going out on a cruise tomorrow. So if that cruise goes out, you really ought to see what's happening to all this water and all this other stuff and go take samples." It's like, okay, fine. So when the stream actually ran down enough that I could get out late that afternoon, I went and packed up my ship pack and talked my way onto the Blue Heron to see the lake completely orange from suspended sediment. And I thought I was going to be able to hand my samples off to somebody else. Look, I got these great samples. Don't you want to study the flood event for the summer? And they're like, oh, yeah, you should study the flood event for the summer and tell us about it. So suddenly, Brandy's and my summers became completely different than what we'd originally planned. But I want to give you a little bit of a background on the angle from which I'm coming, which is that when I'm not chasing the flood, and I hope not to be this year, I'm actually more interested in organic carbon and carbon cycling. So I'm interested in how the landscape interacts with a lake, a stream or a river, or the ocean, and how the organic matter, which I think of as the food source, the base of the food chain, is actually interacting. And if you're in a lake system, you get organic carbon from photosynthesis and chemosynthesis, or from import from the landscape around the lake. And how much of each of these is involved in supporting other things living in the lake depends upon the size of the lake that you're in and the residence time of the water that you're dealing with. So if you have a little tiny lake, the landscape impact's much stronger. If you have a bigger lake, you get very different responses. When you have a flood, you might have a shift in how those balances between in situ or autochthonous production and allochthonous or out of the lake production might be occurring. Now, another sort of bias that I have as someone who cares about carbon is that I'm very interested in how organic carbon and inorganic carbon or carbon dioxide are interacting. And I would argue that we should be too, for a couple of reasons. One is, I like to breathe oxygen. I think most of us like to breathe oxygen too. And we get oxygen from the mismatch of photosynthesis versus respiration. If we respired everything that photosynthesized in a quick coupling, there would be no oxygen in the atmosphere. And in a long-term sense, if you're dealing with oil and gas and shales and mountain building and all those other issues, the mismatch between the burial of organic carbon and the respiration and burning of organic carbon is what sets the oxygen level for our atmosphere. And that's a really nice oxygen level. It gives us enough to breathe, but not so much that we spontaneously combust. And it would take only a few more percentage for us to be a much more combustible species. And I like doing both the breathing thing and the not combusting thing this afternoon. All right, so we have this balance between photosynthesis in this direction and respiration in that direction. You could also think of it as making sugars and biomass and the carbon dioxide mix. All right, a little bit more about carbon cycling and the production of organic matter. We've got some jargon we've got to wander through, so bear with me here. Primary production is the making of organic matter from inorganic starting materials through the process of photosynthesis. It's also called the fixation of carbon. So primary production, your plants, your algae. Organic is stuff that's derivative of hydrocarbons. Carbon, hydrogen, oxygen, reduced carbon species, things that make biomass. Inorganic in my world is carbon dioxide, carbon sulfide, carbonates, graphite, diamond, and cyanides. Now, if you have primary production, you also have secondary production. I am a secondary producer. I eat plants and I make meat, so I'm a product of secondary production, sometimes tertiary and more. I do eat meat on occasion. So anyway, primary production, secondary production. Okay, if we're taking a water system and we're looking at the organic carbon, carbon dioxide, oxygen issue, we have formation, primary production, generally occurring in surface waters in large lakes where there's enough light, and we have them throughout the water column in shorter lakes, shallower lakes, where light can actually reach the bottom and you have benthic production. You have the organic matter that's produced sinking out of the water column and hitting the sediments where there are lots of things that eat it. And you often have, as the things eat it, respiration of carbon dioxide, same way that we do, consumption of oxygen, and if you have a lot of organic matter raining down, you use up all the oxygen, you get an anoxic event. And unfortunately, multicellular organisms require oxygen. Bacteria are really good at using all sorts of different chemicals. They're much more flexible. That's their niche, is that they can deal with lots of different chemical situations, lack of oxygen. But we multicellular guys, we want oxygen, and so if this goes anoxic, you're not going to have animals, worms, other things living in the sediments around them. So if you were looking at the sort of cycle of organic matter, you've got carbon dioxide coming up from the deep waters and the sediments, you've got oxygen produced near the surface waters, you've got rains of organic matter into the deep waters and the sediments, and you also have a temporal component. So here's where the phenology comes in, where oftentimes, if you're a lake or a river, you have nutrient inputs, say with snowmelt or rain events in the spring, and then afterwards you get algal plant biomass using up the nutrients. Especially if you're in a deep water system where stratification occur to trap the plants where there's enough light that they can actually use the nutrients. I just threw a lot at you. Questions? Okay. Let's talk a little bit about food chains. Now, in most food chains, you only make biomass out of about 10 to 30% of the carbon that you eat. We're not very efficient in these things. The rest of it gets respired, gets used to make us move around, lets us do the other things that we do as living organisms. So a typical land food chain might be grass, and then rabbits and foxes. And here's the pyramid of biomass and the pyramid of available energy as reduced carbon bonds. Now, when you're in a water system, there's often a very tight coupling between primary production and secondary production. Because if you have a bloom of algae, very quickly afterwards the zooplankton bloom as well. And in those sorts of situations, you can have less by numbers and less by biomass because there's that tight coupling. As soon as something gets made, it gets eaten. So you can have these little bulges in the water column. But in general, and if you look at the available energy, there's going to be more energy available the lower down in the food chain you are. This is one of the arguments for being vegetarian, actually, in terms of carrying capacity of systems, which I think about, but don't follow. Anyway, primary production needs. If you're an alga and you're floating around in a lake, you need light, you need nutrients, so you need phosphorus, nitrogen, you need a little bit of iron, and if you're a diatom, you need silica to make your little protective shell. And you need appropriate temperatures. Different types of organisms are more fine-tuned for specific temperature levels, and in general, respiration processes increase every 10 degrees C that you warm up a water column. All right, with this set of needs, you end up getting a bloom or extensive production once you start to have an onset of stratification, where you have enough light, where you have enough nutrients, and now you have appropriate temperatures for good growth. So that might be a spring bloom, but then you have the problem that you've used up all your nutrients. And then you may need to rely on nutrients coming up from below or a remixing of the water column. And so that kind of sets the carrying capacity of your stratified water. All right, a little bit about stratification. Here I'm actually going to the Mediterranean Sea. So here the stratification is both a function of temperature and salinity, but more so temperature in this case. And what I'm showing you here is temperature in this sort of dotted set of lines, and fluorescence, chlorophyll fluorescence, in the solid lines. And what you can see is that in May, and then moving into June, July, and August, you're getting ever-increasing stratification. And as you have this increased stratification, you end up developing a subsurface chlorophyll maximum. This is sort of the sweet spot for the algae, where there are enough nutrients coming up from below, and there's enough light coming up from above that they can grow really well. Above this, a lot of the nutrients have been used up already, and the other challenge is that if you have too much sunlight and you're an alga, you end up having trouble with your DNA and RNA and saturating your photosynthetic apparatus, and you aren't a happy cell. So happy cells down here. All right. The other thing to notice is you could pretty much, in the same way that you looked at your pictures and decided times of year, you could look at a water column and figure out what time of year you're in by looking at the temperature profile and the chlorophyll profile for the system. So spring into summer, and then as we get into fall, you get a deepening of the mixed layer because you have a lot of energy from the wind, but not enough to totally mix it, just enough to kind of let it eat its way down into the water column. And then eventually you have mixing that occurs in December. Questions about that? All right, so phenology, if you want to be a real purist about definitions, is dealing with biology. But I've seen people talk about ice phenology, and I like to use it in that less pure sense. But anyway, phenology is the study of periodic plant and animal life cycle events in nature, and they're affected by climate and by seasonal change. And what we're kind of curious about now is we're having climate changes, we're having changes in ice out, we're having changes in temperature levels and when you get higher temperatures. So what happens to the biota when the timing changes? So just to give you a little bit about changes, this is data from Oster and Coleman in 2008, and it's looking at air temperature and water temperature in Lake Superior, and looking at the longest data set that's available right now, which is from Sault Ste. Marie over the past century. And what you can see is that about 1980, we had a bit of a phase change in terms of how the temperatures in the air and the water are reacting. And if you look at the water temperature, it's increasing faster than the air temperature. And this is believed to be a feedback mechanism with ice, because there's less ice cover since the 1980s, and ice reflects light and prevents the light from being absorbed by the water and causing the water to warm up. So if you have an ice cap on the top of the lake, it's going to be pushing all of that sunlight away for a while. And when you don't have that, and you have nice, deep, dark waters throughout the winter, you're going to be able to soak up that heat, and you'll have warmer water temperatures overall. The same sort of thing is happening in the Arctic right now as a sort of feedback mechanism on ice cover there. This plot should look familiar to you guys. At least part of it should look familiar to you guys. This is all three of them plotted together, the central, eastern, and the western moorings. And any one of them you could sort of squint and say, yeah, short, small data set, okay. But the fact that all three of them are showing the same trend from 1980 to 2000 of a decrease over time is a little more troubling. Yes, we have changes. A lot of these sort of periodicities that are every 6 to 10 years have to do with things like El Nino, La Nina, and those sorts of oscillations in weather and temperature. But then if you pull out that sort of information, we are seeing that the onset of Lake Superior stratification is coming earlier in the summer. Now, just to remind you that stratification is important, here's data from M1, which is about halfway up the western arm of Lake Superior if you're heading toward the Apostle Isles and Isle Royale from Duluth. It's in the middle of the lake. You can see the land if you squint, but not really at this site. And what you can see here is that in August, we've got a nice stratified water column, very warm temperatures at the surface. It's up at about 17 degrees Celsius. It's good swimming weather, actually. But you don't want to get down below about 10 meters because it drops right off down to our 4-degree thing, much deeper at about 30 meters. And in this particular plot, we have chlorophyll that's actually up into the surface waters. We don't have that subsurface chlorophyll max here. And this is color dissolved organic matter fluorescence. which is the sort of brown stuff coming in from the streams and rivers. So this sort of gives us a picture of what's going on summer at that site. Now, if we look at changes that have been occurring, we've changed the onset of stratification, which means we're changing the timing when we get production of the algae. And how the zooplankton respond to that is an open question between the coupling of food source and eater, and then also the sort of temperature regimes that the plants and animals can live in is a question. We're also seeing changes in terms of total phosphorus and chlorophyll levels. So here, this is data from 1950 to 2000, and this is from 1970 to 2000. And what we're seeing is that total phosphorus concentrations in the lake are decreasing. And remember that Lake Superior is phosphorus limited. So if you decrease the phosphorus levels, then we decrease the chlorophyll levels too. And this may be a function of a couple of different things. One is that we have cleaned up our act quite a bit. So cultural eutrophication, us burning things and having sewage inputs, that's been reduced. So some of this may be moving back to more natural levels. The other thing that shifts is phosphorus tends to come in with sediments. It comes in as a solid. And so if you have changes in sediment loading, changes in snow cover and snow melt, that can also affect phosphorus levels. So we're not exactly sure what's causing this here. It may be a combination of factors, but we are seeing this trend occurring. The other thing that seems to be shifting in the lake is zooplankton levels. So if you look at the surface, so the sort of surface mixed layer here, we have 2 years. We have August 2010 2011, and these are snapshots, so it's just illustrative rather than completely convincing, I might say. But what you can see is that Daphnia, which can handle warmer temperatures, becomes much more prevalent in the warmer year. And we could be seeing, and we are seeing, shifts in the zooplankton compositions in the lake. And we haven't got enough data to really pin down why or how persistent they're going to be. But how this moves into the food webs could also be very interesting, because fish like us have favorite foods. All right, another thing that's happening, and I stole this data from a colleague, Eric Brown, who stole this data from the Minnesota website of Mark Seeley's. And this, I don't know why he chose 2-inch rain events, but he chose 2-inch rain events. And this is showing you the running mean of 2-inch rain events from 1870 out to present time. And somewhere around 1930, we started to have an increase again in the number of 2-inch rain events. And if you actually go to the same website and look up mega rain events, which are defined as 10 inches of rain over 1,000 square miles in a single event, there have been 10 recorded in Minnesota, I believe, since 1870, and 4 of them have been in the last 10 to 20 years. So we're seeing an increase in extreme events, as well as this increase since 1930 of the 2-inch events. So we're changing how the water is coming in, and this is actually consistent with climate change model results, that we're going to have perhaps less rain overall and more rain in gully washers. All right, so possible ramifications of these sorts of things are, over decades, Brandy showed you that nitrate in Lake Superior has been increasing. The phosphorus I showed you is decreasing, and the chlorophyll is decreasing. And this could be, in terms of the phosphorus levels and the chlorophyll, due to decreased snowmelt and sediment inputs. But the main phosphorus source in Lake Superior is believed to be atmospheric, because Lake Superior is a big lake with a huge surface area, and it has a really small watershed. If you look at the lake to watershed ratio, let me say it the other way. The watershed to lake ratio is 2.5, which is pretty small for lake systems. The other thing is there's better water treatment, there are better emission controls, and there may also be a feedback with the stratification. Because remember I told you the zooplankton composition seems to be changing in the surface water? Zooplankton poop differently, different kinds. Some of them poop in a really diffuse way, and some of them make sinkers. So if you're a copepod and you make sinkers, the phosphorus and the nitrogen that you poop out goes deep into the water column and isn't up in the surface to continue algal growth. And if you're a diffuse pooper, then your nutrients stay up nearer to the surface for longer. So there may be some feedback issues going on there as well. Now, we're seeing a shift in stratification, so it's quite likely that there's a shift in primary production timing. We're working on that right now to understand more about it. We could have a shift in nutrient dynamics, and remember what I said about the pooper, sinker versus other. We might actually be able to mitigate this loss of phosphorus because if cladocerans, which tend to like warmer water, become more dominant, they're diffuse poopers, so that phosphorus would stay up for longer. So we're not sure what's going to happen in this case. And then the shift in food web structures for things like Cisco and Kai is also an open question. Any questions about that? There's a lot to learn, and we're at the very beginning stages. The physics is a lot easier to see the changes to than the biology. All right, whoops. So what I'd like to do now is talk a little bit about that flood event that you already heard about. So you'll get my perspective on it too. In June 19th and 20th, we had a lot of rain, and the rain was mostly focused in the North Shore, Duluth, and a bit farther up. By the time you get to Grand Marais and Thunder Bay, much less so. And if you looked the day after the rain event, what you saw were very, very concentrated, very orange waters near shore from the sediment runoff. And when I looked out from my house into Lake Superior, it looked like Lake Superior was this vivid, vivid tang color orange, which was pretty dramatic. But, you know, I live here, so I wasn't seeing the open lake picture. Now, as you move later and later, that really concentrated sediment plume is now mixing into the western arm, the far western arm. And what we did for our summer's work is we monitored these 3 sites every week, and we tried to get to these sites every other week for summer through the fall to see what might happen. Initially, we had hoped that this OS site would be the edge of the flood, so we could compare OS to these two and we wouldn't have to go on the ship so long. But we were out of luck. The flood was bigger than we expected and affected a lot more of the lake. So, offshore site, Duluth entry site, Wisconsin entry site for the rest of the talk. Up here what I'm showing you is total phosphorus, and total phosphorus skyrocketed right after the flood event, most likely because all the sediment came out and phosphorus likes to stick to particles. If I looked at soluble reactive phosphorus, it also increased. Total phosphorus increased 100-fold. Soluble reactive phosphorus increased 5-fold in the flood impacted waters. But that spike did not persist for very long. It lasted for a week, maybe 2 weeks at most, and then disappeared. And if we look at chlorophyll concentrations never responded, which is absolutely, positively not what I had expected to happen. And to give you another view of this is a picture of total suspended solids to give you proof it really was the solids here. So what we did here is we collected the solids onto glass fiber filters, and then after we measured the change in weight for the dried solids on the filters, we also took those filters and we combusted them. So total volatile solids is what you can burn off the filter, which is a proxy for organic material. So the stuff that came out was pretty inorganic, so the organic's pretty small here, all the way through in the red, and we had a lot of it. This is milligrams per liter. We had almost 100 milligrams per liter of sediment in the lake right afterwards, but it dropped off by the beginning of July. Now, notice the scale change. 100 milligrams, 1.8 milligrams. So by the time we're out into August, we're back down to normal levels of 1 to 2 milligrams of suspended sediment at the sites. And notice also that the percentage that's organic is now up quite a bit. Now the particles are much more organic. It's a lot of phytoplankton, biota, living things in the water column rather than the washed out sediments. I think you saw this earlier. So this is dissolved organic carbon. These are pre-flood values, about 2-ish milligrams carbon per liter. Post-flood, where we had our impacted sites, remember OS we thought was going to be the clean site. This was the first day. So we had roughly 6 milligrams per liter carbon at our impacted sites, same 2.4 at the unimpacted site, but then things started to mix in. And the DOC didn't drop off the way we expected and saw that the total suspended solids did. And this gives you a sort of visual picture of that. And if I plotted Secchi depths, do people know what Secchis are? So you take a disk, it's got black and white markings, you stick it in, you say, oh, I think I see it still. Oh, no, I can't. That must be the light penetration depth. Okay, we did Secchi as well as these sorts of measurements. And the Secchi depths got really shallow after the flood, and they stayed really shallow for a while afterwards. So remember that M1 site I said was halfway between, and I can show you. M1. All right, so pretty far into the lake. At the M1 site, this was our non-flood year, August 18th. Nice stratification, chlorophyll fluorescence, no major amounts of chlorophyll below the thermocline, CDON fluorescence. When we moved to July 20th post-flood, we had nice stratification, but we had a huge colored dissolved organic matter fluorescence signal that was trapped in with that warm water. And we had a suppression of chlorophyll. So between the Secchi depth and this data, what looked like was happening is that we had this lens of brown water at the surface of the lake, and it was preventing light penetration, and it was preventing algal growth. And if you actually looked at the lake visually, the lake looked this really odd purple color, which was sort of a blend between the brown of the stream water coming in and the blue that you would normally expect to see. So, based on all of this, I can say that the lake is changing due to interactions with climate, land use, and anthropogenic inputs. We did see a strong immediate flood response in terms of total suspended solids, DOC, and nutrients. And then we saw some suppression of the primary production. In terms of longer-term effects, they seem to be pretty moderate. I'm not sure that we'll see a huge difference when we go out this year in terms of algal productivity and light levels or nutrients, but we're going to go check and see. Now, what this might do in terms of fish may be different, because a lot of the fish have as their nurseries the sort of rocks along the shoreline. And we pretty much threw a lot of sediment on the shoreline. And then the other thing we did for those fish that like to spawn upstream is we pretty much reset a lot of the stream beds, at least in our area. So how those sort of effects are going to play in, we don't know yet. It's likely that the sort of flood event that we saw is going to come again. We're seeing a shift in water inputs, increasing extreme storm events in our region. We're seeing a bit of a shift in when water is delivered as well. And for future work, we're going to be doing water quality sampling this summer at a lot of the same places in the lake. And we do have at the western mooring site, the same place that you drew your graphs of the data, we have sediment traps, which are these things here, that have been out for about 10 years and will stay out, and they collect what rains into them from the surface waters. And we can measure the organic carbon in them and get an idea of productivity from year to year. And what's kind of neat is, I don't know if you can see down here, but there are little bottles that get rotated every 10 days or 14 days, so we can get a time series and then pull the traps once a year. We're also quite curious to look at the data that we have now, along with historical data. So if you guys know of any historical data sets, please let us know. They're very, very useful pieces of information. And I think you might have met this person. Here's a picture of grad student Brandy Forsman measuring color dissolved organic matter on the RV blue heron last summer. And I wanted to point out that what I'm showing you today is not all my own work. It's work from lots of people who donated time, sample bottles, effort. We kind of did this flood response work on a shoestring. Although we did have some nice funding from Minnesota Sea Grant and also from the Great Lakes Observing System that helped us pay for some of our ship time. And lots of people helped with the sampling and analyses.